My strengths lie in computation, so I decided to observe the cell's behavior virtually. And simulations are designed after loads of computer programming and entering available information from real life experiments. So we searched for theoretical models that would explain what Angelini observed in the petris. We found a very basic model called Cellular Potts Model. This model, though simple, can capture key characteristics of how cells actually move in the single layer. Also, all this time, not many studies explored the glass-like dynamics of cells using this model. But what is the point of these simulations? These simulations helps us play around with the different conditions and then observe how these different conditions affects the layer of cells. Also, they help visualizing a situation over a really long time scale, something that is not achievable in real life. Now, Angelini's experiment said that if the density of the cell layer increases, the cells gradually stop moving. But I couldn't increase the density of the virtual cells. They are already stuck with each other and covered the entire virtual field. So what I decided, I looked at other ways to influence the movement of the cells. One of the ways I could affect the movement of the cells is via regulating their activity. The more active the cells, the more they move. Now in the simulation, one of the things that regulates the activity of the cells is the temperature. So I decided to play around with the temperature in my neat virtual experiment. So I observed that decreasing the temperature lead to a slowing down of cell movement. Now that we found a way to influence the movement of the cells, we decided to observe the cells and spot glass-like dynamics in the cell layer. So over time, I observed something interesting in the simulation. A group of cells restricted the movement of one cell by forming a cage around it. Molecules in glass are similarly caged by their neighbors and thus coming to a standstill for a long time. I also found that certain group of cells moved with a specific velocity. Some of the groups move faster than the others. And this feature is called dynamic heterogeneity, which is a hallmark of glass.